Just for a quick overview, let's just go over again what we talked about chromosomes. We know that in our normal cells we have chromosomes we got from our mom and chromosomes we got from our dad. So if we're talking about one particular chromosome, like chromosome number 1, we got one from mom, one from dad, we call that a pair of homologous chromosomes. Don't forget that we get one chromosome from each parent. That is a homologous chromosome. They each have the same set of genes that they're going to code for, except they have different versions of them. Another quick overview, diploid meaning two sets. Diploid cells have two sets of chromosomes, one from each parent, which we're already familiar with. We also know that these cells that are diploid are going to be all of our normal body cells. This would be our somatic cells. So make sure you know that word right there. Cells that do not have two sets of chromosomes, since there's only one type of cell that's like that, is our haploid cells. Our haploid cells have one set. That means if you're human, your haploid cells will have 23 chromosomes, and there's only one type of cell in our body that will have a haploid number of chromosomes, and that is our sex cells or our gametes. Karyotype. What exactly is a karyotype? All a karyotype is a picture of all the chromosomes in an organism cell, and they're usually arranged by size. So if we look over here, what we're looking at is a human karyotype. We know we have a total of 46 chromosomes, 23 from our mom, 23 from our dad, and we pair up the homologous chromosomes in a karyotype, arrange them by size. Very last pair 23, will be your sex chromosomes. So why do we need to know about the life cycle of cells? Well, all cells aren't always dividing. Every cell has a different lifespan. So if you look at this chart, you can see, depending on what kind of cell you are, you might live a very long time or you might live a short time, and you may be constantly dividing or you may never be able to divide. If we look over at our digestive tract cells, right up top, you see that most of them have a lifespan of less than about 10 days. We're constantly using our digestive tract straight from our mouth all the way down to our intestines, so we can see that these cells are going to be constantly dividing. Our red blood cells are replaced about every 120 days. That's why we can donate blood. Our white blood cells are a little bit different. We have different types of white blood cells. Some white blood cells are going to stay in our bodies forever, holding important information about how to fight certain infections or viruses, bacteria, and these are what we call our memory cells. So they can live a very, very long time. However, we have some short-lived cells that when you get sick, the cells start dividing, they fight off the infection. After that, they're done. So they can live about 10 hours. The life cycle of cells consists of two major stages. The first major stage is what we call interphase, and that consists of three different parts. We have G1, S, and G2. And you can look over here and see what's going as far as those different stages. If we come over here, every cell is going to be spending time in G1. If your cell's just doing what it's normally supposed to do, it will be in G1. This is where our cells start off. They might get a little bit bigger, the cytoplasm gets a little bit bigger, but we've talked about before how important it is to keep that surface area to volume ratio high. In order to do that, we need to make sure the volume stays small, but we increase the surface area. So again, the cell will grow. If it's done growing, okay, it'll come out, and sometimes we say that it's in G0. And that's just going to be considered when the cell's doing its normal job, when it's pumping out proteins and all that good stuff. Now let's say we're talking about one of the cells in your arm bone. So if this is one of the cells in your arm bone, it would be in G0 because you're fully grown, doesn't need to divide anymore, and it's fine. However, if you happen to break your arm, that cell's going to now have to divide. So then it reaches this R checkpoint, which decides whether or not the cell is going to divide. If your arm broke, the cell knows it needs to divide. It gets certain signals, certain growth factors. So it's going to start the process of cell division, or get ready to start the process. That's when we enter the S phase. The S phase is where our DNA is replicated. This is a really important aspect. We drew that out on the board. Okay, remember, normally in your S phase, we wouldn't really see the individual chromosomes. It would actually be a big tangled mess, and these big tangled mess of chromosomes would all replicate. And then once they replicate, we also have our, if you come over here, our centrioles. We normally have one pair. During S phase, our centrioles replicate. After we leave S phase, we enter into G2. The G2 phase is where the cell is going to make its final preparations to divide. all of that, this whole entire aspect here, is what we call interphase. So interphase is 3 phases, G1, S, and G2. Most cells are going to be in G1. If they're fully grown, if they're not dividing, they stay in G1. Sometimes refer to it as G2. For whatever reason, if the cell needs to divide, that's when it's going to move into S phase. Again, there's a very important checkpoint right here that controls that. S phase, DNA replicates, DNA synthesis, S for DNA synthesis, DNA replicates, centrioles replicate, and then we move into G2, where the cell is just preparing to divide. That'll take us to the second stage of the cell cycle, and we call it M phase. And M phase consists of 2 parts. M phase consists of what we call mitosis, which is division of the nucleus. We see that over here. So mitosis, division of the nucleus. And we're going to end up actually with one cell that's going to have 2 nuclei in it. And we know we can't have that. So eventually we're going to have to get into the second part of M phase, which is what we call cytokinesis. Remember, cyto means cell, and kinesis has to do with movement. We're actually going to be splitting the cytoplasm. So this would be the process of cytokinesis. This is what you would see. The cytoplasm, the nuclei had already divided, and now the cytoplasm divides until eventually we end up with these 2 identical cells. So that's a brief overview of our cell cycle. Again, we have 2 major phases, interphase and M phase. Interphase is divided into 3 stages, G1, S, and G2. We only get to the S phase and G2 if the cell is going to divide. Once the cell leaves G1, once it goes past this checkpoint, it's committed. That means it is going to divide. Okay? The only way it will stop dividing is if something happens along its journey to get into M phase. DNA doesn't replicate like it's supposed to. The centrioles don't replicate. Anything like that is the cell will self-destruct. And we talked about what that was. Apoptosis, which is when the cell self-destructs. And again, this is a good diagram. This is what you did in your coloring. We're looking at the entire cell cycle. So this over here is what we call the cell cycle, or the life cycle of the cell. And we have interphase going all the way around. We know G1, it's doing its job. And then once it gets a certain signal, right here is where we're going to have our checkpoint. And it's really important for you guys to know that and to understand that in order to proceed to the other parts of the cell cycle, to continue on to M phase, it has to reach this checkpoint. It has to get the right signals, the right proteins to go on to the next stage. Once the cell goes on, it is committed. It will finish the cell cycle. S phase, DNA replicates. Make sure you know that. And then G phase, we prepare to divide, and then we enter M phase. And you can see M phase is divided into 2 stages. The first stage is what we call mitosis, again, division of the nucleus, and then finally our cytoplasm splits, and we end up with our 2 new daughter cells. What is actually controlling all this? Okay, we have internal and external factors that control the cell cycle, which we're going to get to. And I just want to point out in this picture, these are some of our internal factors that the cell uses to control how it goes from G1 to S to G2. And we call them cyclins and cyclin-dependent kinases. And all you really need to know about that is they're really important proteins. And this one should give it away. It's a kinase, ASE. ASE means enzyme. So CDK is a cyclin-dependent kinase, and then we have our cyclins, which is just another protein, which is going to be our substrate. So if this is our cyclin, this would be our cyclin-dependent kinase. And so this guy would end up attaching over here. Once they're attached, the cell will proceed into the next stage. The most important thing to remember about interphase is that there's many checkpoints found throughout. In an interphase, there's a checkpoint in G1, there's a checkpoint in S, there's a checkpoint in G2. The importance of these checkpoints is to make sure everything in the cell is ready to go. make sure that the DNA is replicated properly, that there aren't any mistakes. If there are mistakes, then the cell will not go past that checkpoint and it will go through apoptosis. Many cells spend most of their life in G1 because they're non-dividing cells. So those would be such as our muscle cells or our brain cells. Like I mentioned before, we have different types of regulators for our cells. We talked about those internal regulators, which were our cyclin-dependent kinases and our cyclins, and those again are just proteins, fancy names for some important proteins in the cell cycle. And we have a couple of them. We have many different types, and you can see up here, this is one CDK, this is another one, and they're going to play very important roles in controlling how the cell moves through this process. We refer to them as internal regulators of the cell cycle. They control everything from the inside. So just keep that in mind. You'll never really be asked a question as to what is a cyclin-dependent kinase, but it might ask you, how does the cell regulate the cell cycle? So you need to be able to differentiate between those internal regulators and then the next thing that we'll talk about, which is what we call external regulators. External regulators are things outside the cell that are going to control the cell cycle. For example, when you break a bone, at the two ends of the bone that are broken, the cells will begin releasing proteins that will signal to the other cells across from it to start dividing. Those would be external regulators. So they're factors outside the cell that tell other cells around it to start dividing. Growth factors is a very good example of how external regulators get cells to go through cell division. Another important external regulator is what we call contact inhibition. And what contact inhibition has to do with is that when cells are dividing, we can look over here and imagine these are the two ends of your broken bone. They release external signals, external proteins to tell the cells across from it to start dividing. Once they come in contact with each other and there's no longer a need to divide, okay, contact inhibition will occur and the cells will no longer divide. However, if those cells, for whatever reason, do not have their external signals working properly, such as those membrane proteins, extrinsic or intrinsic transmembrane proteins, if those proteins aren't functioning properly and they don't recognize a signal from contact inhibition to stop, then they will continue growing out of control, and that's when we get what we call cancer, which we'll get into later. All right, so now we're going to dive into the second major part of the cell's life cycle. We already talked about interphase. Now we're going to look at M phase, and this is where the cell is actively dividing, and it consists of two different stages: mitosis followed by cytokinesis. Again, mitosis is division of the nucleus, and cytokinesis is division of the cytoplasm. And finally, when cytokinesis is over, that's when we have our two new cells. An easy way to remember the stages of mitosis is PMAT, a little acronym to remember the 4 stages in the proper order, and we'll go over what goes on in each of these stages. So let's go ahead and look at prophase. When we look at prophase, okay, the first thing that you need to know is that those DNA molecules that were all spread out as chromatin, they replicated, but they were still a big tangled mess, suddenly become condensed and visible, and that's when we can actually see them. So chromatin becomes chromosomes, the nuclear envelope will start to break down, that's represented over here by these broken down orange lines. The nucleolus will actually completely disappear, so we won't see that anymore, and then we're going to see our centrioles start to move to opposite ends of what we would say would be the nucleus if it were still there, but also opposite ends of the cell. And when these centrioles are moving to opposite ends, we call the region where we find the centrioles a centrosome, and that's pretty important to know because we can use either word. We know that plant cells don't have a centrioles, so they have what we call the centrosome, and this is an area where we start having microtubules coming together to make what we call a spindle apparatus. And all that is an area that's going to send out microtubules to attach to the centromeres, which we'll see happening later on. And if we come down here, okay, this is an actual micrograph, and you guys will be looking at some onion root tip cells and be trying to identify different stages of mitosis. All these cells here, and we'll go ahead and get a pen so we can actually see what's going on a little bit. So all these cells over here, if we're looking here, these are the nuclei, and these nuclei, the ones that I'm circling right now, are all in interphase. And the reason we can tell they're in interphase is because if you look in the nucleus, see how it's very, very grainy? That's all of your big tangled mess of DNA as chromatin. Now when we come over here, we can actually see the nuclear membrane starting to break down, and then inside you'll notice that's not as much of a mess as it was over here. We can actually start to kind of make out some chromosomes coming together, okay, condensing. We can make it out a little bit. So this would be early prophase. This is the very beginning. Now at the very, very end of prophase, we'll no longer see any nuclear membrane or envelope. That's completely gone. We'll also notice that all these chromosomes, or all that chromatin, is now visible. And so we now can officially call them chromosomes because we can actually see the individual chromosomes. They're not organized in any particular way, but it's very different than these cells here, 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 okay, and even our early prophase. So make sure you know how to identify, okay, early prophase, late prophase, and the major events that happen. Now I want you to pay special attention to this, okay, what we have at the end of prophase, because when we get to telophase, it's the exact opposite. So keep this in mind. You can always flash back to this slide. If you want to review, I'll have it again on the telophase slide. All right, so after we finish up with prophase and we have those nice visible chromosomes, we're going to enter into what we call metaphase. And like I told you before, remember metaphase, M, middle. That's an easy way to remember it. Metaphase is where our sister chromatids our sister chromatids are still attached to each other, are all going to line up in the middle of the cell. And that's, again, why we get the name metaphase. And as they line up in the middle, what we're going to see start to happen is these microtubules from these centrosomes, where our centrioles were, are going to start to extend. And these are proteins. So again, another really important job of proteins is to make up these microtubules that are going to attach to each centromere. And that's essentially what happens in metaphase. Now, as far as how long metaphase takes, it varies depending on organism. Sometimes it can take 10 minutes, 20 minutes, 30 minutes. It's different. Prophase is usually the longest. 50 to 60% of mitosis is spent in prophase. So we'll usually be seeing a lot of cells in prophase. Metaphase, not as long, but still a good amount of time is spent in metaphase. So the third stage of mitosis is what we call anaphase. And I told you, just easy way to remember anaphase, A for anaphase, A for apart or away. This is where the sister chromatids get separated and they get pulled apart. So once they lined up in the middle and those spindles attached, they're going to go ahead and yank them apart. And this is really important because this is going to happen very quickly. Okay, this stage happens very quickly. So when we're looking at our slides trying to find cells going through anaphase, it's going to be a little bit more difficult because they don't spend as much time in this stage of mitosis. So these sister chromatids are pulled to opposite poles, and then they're going. Then as soon as this is done, we're going to get into our next phase, which is telophase, and our last stage of mitosis, but not the end of cell division. The last stage of mitosis is what we call telophase. And I told you guys this is the exact opposite of prophase. So everything that happened in prophase, okay, we do the opposite over here in telophase. We're going to see that the nuclear membrane is actually going to start to reform. So when we look up top in the top right hand corner, if we look at our nuclear membrane, you can see that it's starting to reform over here around each set of chromatids on the opposite poles. We're also going to start to see the nucleolus reappear because it wasn't there the whole time mitosis was going on. It's going to reappear. We're also going to see the chromosomes that were so visible before start to spread out and tangle themselves back up into our chromatin. And then finally, we're going to see our spindle apparatus is going to break apart. Our centrioles are still there, but those spindles and microtubules are no longer there. Now, at the same exact time telophase is happening, cytokinesis is also occurring. So even though all this is going on here and the nuclear membranes reforming, the cytoplasm is actually getting ready to split up. And so they happen at the same time. we'll be able to recognize this fairly quickly. So cytokinesis is division of the cytoplasm, and when we look at cytokinesis, it's going to differ in animal cells and plant cells. When animal cells cytoplasm divide, we know that animal cells do not have a cell wall, so therefore we don't have to make a cell wall. All that has to happen is our cytoplasm needs to split, and that's exactly what occurs. What we get as the cytoplasm begins to pinch off is something we call a cleavage furrow, and I'm just going to backtrack really quick just so you can see that, because this picture demonstrates the cleavage furrow fairly well. This little area where it's pinching off, let's go ahead and get a pen going. In this little area where it's pinching off right here, this is what we would call our cleavage furrow. So you want to know that term. That's an easy way to differentiate between cytokinesis in an animal cell and a plant cell. Animal cells have a cleavage furrow. We don't have a cell membrane. Okay, so now looking at plant cells, when we want to see how they go through cytokinesis, they're going to have to begin to form their cell wall. Quick review, what's the cell wall made out of? Okay, the cell walls are made of cellulose. Really important for you guys to know that. In order for the cell wall to be made, we have these two new cells are going to begin secreting some of those carbohydrates and polysaccharides, and they make something we call a cell plate. Eventually that cell plate will form around each of these cells, forming their cell wall. So that's a big thing differentiating cytokinesis in animal cells and cytokinesis in plant cells. So make sure you know to look for that cell plate when we're trying to find a plant cell going through cytokinesis and telophase. So this basically sums it up. This picture here, now you should be able to identify each stage of mitosis going on in this picture. And it's pretty easy because it's kind of in order for you. These are plant cells that we're observing here. You can see in the first couple, number 1, 2 are going to be cells in interphase. So they're going to be in G1, S, G2, DNA replicated, and you can actually kind of see the DNA replicating in these cells here. You can see in picture number 1, okay, not as dense, and if we come over here, we actually see that tangled mess is a little bit denser. So this means this would most likely be in G2. This was probably in G1. So then we enter into prophase. So these here, these 3, 4, and 5, these would all be cells in prophase, and you should be able to identify which ones are in early prophase, and then this is late prophase. You can really see those individual chromosomes. Then we come down here, and this is a really good example of metaphase. Okay, both of them, this is where our sister chromatids all align in the middle. What we really don't see is that these spindle fibers were actually coming over and attaching, and they're coming over from the centrosome region, from where the centrioles were. So they've attached, the place where they attach, it's called these centromeres. So they attach the centromeres of each sister chromatid. That's what we have going on over here. And then we're going to enter into, and again, this is metaphase, don't forget, then we're going to enter into anaphase, and that's in number 8. Anaphase, they're being pulled apart. So those spindles are actually shortening, which is really important that proteins are able to shorten themselves, move, all of that. They're actually being pulled in opposite directions. And then we enter into our telophase. Now remember, telophase and cytokinesis happen at the same time. And it's a little bit harder to see what's going on here, but what we're going to start to see happening is our nuclear membrane is going to reform, nucleolus reappears. You can go ahead and draw that out on your own. And as that's happening, what we'll end up seeing is a cell plate begin to form until eventually we have our 2 new cells. And you can see 2 new cells. We no longer see our sister chromatids, or we no longer even see our condensed chromosomes. Now it's a big tangled mess of DNA and another big tangled mess of DNA. And now we're back to looking like this cell up here, our original parent cell, with the big tangled mess of DNA. And that about does it for our lesson on the cell cycle and mitosis. I know this was a lot of information. Why don't you go ahead and practice identifying those stages of the cell cycle in the picture on the screen, and then we'll have a quick review in class when I see you next week.